Welcome to Bioinformatics Introduction and Methods from Peking University. Let's continue with Introduction and History of Bioinformatics. In the last unit, we learned about what is bioinformatics. Bioinformatics is an interdisciplinary field that develops and applies computer and computational technologies to study biomedical questions. In one role, bioinformatics is a powerful technology to manage, query, and analyze big data in life sciences. In another role, bioinformatics is a top-down, holistic, data-driven, genome-wide, and systems approach that generates new hypotheses, finds new patterns, and discovers new functional elements. When you take a course, as you learn the detailed knowledge and technologies, it is equally important to gradually establish a conceptual framework of the field. Once you have established the conceptual framework, the content you learn is no longer isolated fragments, but rather a living, organic whole. In the last unit, we talked about how to look at bioinformatics research from the angle of biology, from the main theme of the central dogma and axis from genotype to phenotype. We also talked about how to see bioinformatics from the angle of information sciences, from the main theme of from data to discovery. I hope that you've got a spatial conceptual framework of the field of bioinformatics. In today's unit, I hope to help you establish a temporal conceptual framework of the field of bioinformatics from the historical perspective. A good field is not static, but rather alive and growing. Once you really understand a field, you feel that you can almost feel its pulse, see its growth. You may even be able to use the present to predict the future development of the field. History is our best teacher. In today's unit, I'll review with you the important events and papers in the history of bioinformatics. We shall learn how the scientific questions were formulated and how the technologies were developed. The historical setting of the formation and growth of bioinformatics is the formation and growth of molecular biology and computer sciences since the 1950s. In 1950s and 1960s, many critical concepts and technologies in molecular biology were established. At the same time, the core technologies of electronic computers and the Internet were formed. In 1970s and 1980s, molecular biology and computer sciences experienced fast growth. The concepts and technologies matured. Since the 1990s, both had experienced rapid growth. One of the most important discoveries in molecular biology is the discovery of the double helix structure of the DNA by James Watson and Francis Crick in 1953. This one-page paper in Nature established DNA as the genetic material. Also in the 1950s, the first protein sequence, that of insulin, and the first protein structure, that of myoglobin, were solved. These works started the golden age of molecular biology. In early 1960s, a few dozen different proteins were sequenced. A few pioneers realized that hidden in these protein sequences might be important evidence for evolution of species. So Linus Pauling from Caltech and Emily Zuckerkandel, who visited from France, collaborated to publish two seminal papers in 1962 and 1963 that reconstructed the evolutionary history of species from analyzing the protein sequences of hemoglobins from different species. In 1965, they published a 70-page long seminal paper entitled Evolutionary Divergence and Convergence in Proteins, and proposed the concept of molecular clock. This together with other works such as the neutral theory and the near-neutral theory by Kimura and Ota, greatly advanced molecular evolutionary biology. Even though today there is a lot more DNA sequencing done than protein sequencing, it was not until 1977 when Fred Sanger published his Sanger DNA sequencing paper. But once it is published, it was quickly adopted because of its significantly higher efficiency than protein sequencing. Thus, in the 1980s, large number of DNA sequences were sequenced. In 1988, another historical event pushed DNA sequencing from single gene level to genome level. That is the famous Human Genome Project. The Human Genome Project was initiated by the United States Department of Energy and later led by the National Institute of Health, with contributions by the United Kingdom, France, Germany, Japan, and China. It had a total budget of $3 billion US. Its aim included not only the sequencing of the human genome, but also the sequencing of the genomes of many model organisms. It also supported the development of many genome-wide high-throughput technologies. The growth of the field of bioinformatics is intimately associated with the Human Genome Project. Bioinformatics and Human Genome Project fueled and supported each other. You may know that Francis Collins led the government effort of the Human Genome Project, and that Craig Venter led the industry effort at Celera Inc. You may or may not know that James Watson is one of the founders and designers of the Human Genome Project. These photos were taken when Dr. Watson visited our Center for Bioinformatics at Peking University in 2007. This gentleman, Dr. Maynard Olson from University of Washington, is a member of the U.S. National Academy of Sciences and also one of the early founders of the Human Genome Project. He led the discovery of the YAC and STS, which were important technologies for genome sequencing. Dr. Olson had kindly filmed a short video for this MOOC, giving us his personal account of the role of bioinformatics in the Human Genome Project and his advice for students of bioinformatics. In 1991, shortly after the Human Genome Project started, Nobel Prize winner Walter Gilbert wrote a short essay in Nature. He suggested that as the genome was getting sequenced, biological research will experience a paradigm shift. A biologist should start with analysis of the whole genome to generate a hypothesis, only then going into the lab to test it. But the Human Genome Project was criticized by a lot of people in the early years. There were many reasons, one of which was that many people looked at the sequencing technologies at the time and decided that the challenges of the human genome sequencing was too great to be finished in a reasonable time frame. Indeed, it was not until 1995 when the genome of the first free living organism was sequenced by Craig Venter and team. The genome was 1.83 million base pairs long, sequenced with the clever shotgun sequencing approach. In the next few years, the genomes of E. coli, yeast, C. elegans, and Drosophila were sequenced. In 2001, the exciting moment finally arrived. The draft sequence of the human genome was published in Nature and Science concurrently. The complete sequence was published in 2004. In 2005, the 454 sequencing technologies started the era of next-generation sequencing. Today, next-generation sequencers such as Illumina HiSeq can sequence multiple individuals' whole genome in one day with as much as $3,000. This technology can also sequence the transcriptome and DNA methylome at genome scale. Every 5 months, the amount of new data generated is the same as the total amount of all the data generated in all of previous human history. This brings unprecedented historical opportunities for life science research. In these 60 years, when life science developed rapidly, computer technologies also revolutionized. Today, laptops, cell phones, and internet are ubiquitous. It is easy to forget that the first electronic computer, ENIAC, was not invented until 1945. One significant milestone in computer science was the information theories published by Shannon in 1948. In the 1940s, computers were so expensive that they were used only in the military and large government institutions. It was not until the 1950s when computers were mass-produced and commercialized. Also in the 1950s, the theory of games, computer grammars, and the first high-level language Fortran were invented. The 1960s and 1970s saw great growth of the computer technologies. In 1969, ARPANET established the first connection between Stanford University, UC Santa Barbara, UCLA, and University of Utah. In mid-1970s, the concept of today's Internet and TCP protocol were invented. In the 1980s, personal computers such as those manufactured by IBM were commercialized, finally making computers available to work and home alike. In 1991, the World Wide Web and the Linux system were available. The Internet era began. The creation and growth of bioinformatics as an interdisciplinary field of life sciences and computer sciences is an inevitable historical trend. Some of the classic bioinformatics questions first emerged around 1960s. In the 1980s, the scientific questions, technologies, and researchers reached a critical mass, and bioinformatics as a field emerged and experienced astonishing growth since the 1990s. The first appearance of the word bioinformatics was in 1970 in a little-known Dutch paper published by Hesper and Hoogeweg. They defined bioinformatics as the study of informatics processes in biotic systems. In 1978, Pauline Hoogeweg wrote in an English paper that she defined her research as in bioinformatics. Many people refer to this 1978 paper as the origin of the word of bioinformatics, but the word actually appeared earlier. Before the word bioinformatics even appeared, there were already several true pioneers. The earliest work that I could find that fits our current definition of bioinformatics is a computer program for the determination of myoglobin protein structure developed by John Kanju and collaborator. This work was first presented in 1951 at the Second Computer Science Conference in Manchester and published in Acta Crystallography in 1952. More and more protein structures were solved in the next 20 years. In 1977, the PDB database were created to store protein 3D structures. At the same time, people started to predict protein structure using computational methods. A well-known early work is the Tau-Fessman secondary structure prediction method in 1974. In 1975, Michael Levitt and Walsall published their historic paper on computer simulation of protein folding, which had led to a Nobel Prize in chemistry in 2013. To provide a systematic and fair evaluation of the increasing number of prediction methods, John Moult and colleagues started CASP, Critical Assessment of Protein Prediction, in 1994, which had been running for 20 years. Sequence analysis has always been a central problem in bioinformatics. Hidden in the sequences is information about the function, evolution, and regulation of genes and proteins. The earliest work is the COMP-protein algorithm developed by Margaret Dayhoff and Robert Ledney in 1962. Although protein sequencing technologies were invented in the 1950s, up to 1962, only 6 relatively complete protein sequences were solved. The proteins were broken into little pieces and the sequences of the pieces were determined. Then the pieces were assembled into a complete protein sequence. As Margaret Dayhoff pointed out in her paper here, the assembly problem is a logic and combinatorial problem that requires a team expert a long time to accomplish. The longer the protein is, the more challenging the assembly is. If computers can be used to do the assembly, it may enable more biologists to solve more and longer protein sequences. COMP-protein is the first ever computer program for sequence alignment and assembly. It was run on the IBM 7090 at Johns Hopkins University. Margaret Dayhoff did her undergrad in mathematics and PhD in quantum chemistry. She was working at the National Biomedical Research Foundation in Maryland when she published the paper. She is a true pioneer in bioinformatics, making important contributions in several fronts. To give another example, in the 1960s, she realized that more and more proteins were getting sequenced. So she created a collection of protein sequences. Since 1965, she would publish an annual atlas of protein sequence and structure. In the first year, it contained all 65 protein sequences that were known up to that time. This was the first bioinformatics database. In 1983, the atlas evolved into the Protein Information Resource, PIR, which eventually was taken over by UniProt. Starting from the first 65 protein sequences in 1965, there are 43 million protein sequences in 2013. In 1982, the GenBank database was created to store nucleotide sequences. It had grown from 606 sequences in 1982 to 300 million sequences in 2013. It has been growing exponentially, doubling every 20 months. Margaret Dayhoff not only developed the earliest sequence analysis computer program and sequence database, she was also one of the first people to use computational methods to study the evolution of protein families. She published a paper in Science in 1966 on the evolution of the ferredoxin protein family. The mid to late 1960s were a golden age for molecular evolutionary biology. Linus Pauling published a famous paper on molecular clock in 1965. In 1967, Walter Fitch and Emmanuel Margoliouth published their paper in Science on the construction of phylogenetic trees using computational methods. According to a recollection by Walter Fitch later, he was an assistant professor at the University of Wisconsin-Madison in 1964. He saw the importance and feasibility of constructing phylogenetic trees and started to write an algorithm and computer program for it. He chose to study cytochrome C because it had a total of 10 known sequences, the most among all protein families. In 1966, Emmanuel Macoliash visited University of Wisconsin. He mentioned that his lab had 10 more unpublished cytochrome C sequences. So the two collaborated to build a phylogenetic tree of cytochrome C using the 20 sequences. The result was this classic paper. As more and more protein sequences were determined, it had become a pressing question to evaluate the similarity between two sequences. Such an evaluation requires aligning the two sequences. The number of sequences were quickly exceeding the capacity of manual alignment. In particular, when two proteins have low similarity, it is challenging to find their optimal alignment. In 1970, Saul Needleman and Christian Wunsch published their seminal paper in the Journal of Molecular Biology. They used a dynamic programming algorithm to quickly find the optimal alignment. One of the most important parameters in sequence alignment is to evaluate the goodness of fit of a pair of aligned amino acids. In 1978, Margaret Dayhoff systematically analyzed the substitution rate between different pairs of amino acids among homologous proteins and constructed the famous PAM amino acid substitution matrix, providing an elegant solution for this critical question. The PAM matrix were the default matrix for sequence alignment for many years, even after Dayhoff passed away. Even after the Blossom matrix were published in 1992 and became the new default, PAM matrices continued to be used till today. By the end of the 1970s, introns and intron splicing were discovered. Needleman-Wunsch's global alignment algorithm could not adequately handle the alignment of two DNA sequences when both exons and introns existed. So in 1981, Temple Smith and Michael Waterman published their famous 3-page paper in the Journal of Molecular Biology. It reported the famous Smith-Waterman local alignment algorithm that could find the most similar pair of subsequences, providing an ingenious solution to the problem. I'm pleased to tell you that Dr. Michael Waterman had filmed an interview with us for this MOOC, telling us his personal story of how he had developed this important algorithm. I hope that through discussion of the history, we can teach you not only the details of the methods, but also how to identify a biological problem and solve it using bioinformatics. Maybe the next historic bioinformatics methods would be developed by you. In the 1980s, more and more protein and DNA sequences were solved. A new problem emerged. Biologists need to know whether any of the known sequences were similar to the ones that he was studying. Up to December 1989, the number of known nucleotide sequences reached 31,000. That was big data back then. Needleman-Wunsch and Smith-Waterman algorithms were too slow to handle such a large number of sequences. In the 1990s, based on the statistics worked out by Sam Carden, Steve Altru et al. Published the historic BLAST algorithm in the Journal of Molecular Biology. By ingenious indexing of the known sequences and sacrificing optimal alignment in some situations, BLAST increased the speed of database search by order of magnitude. In 1997, they published the gapped BLAST and SciBLAST algorithm in nucleic acids research. According to Google Scholar, both of these papers had been cited nearly 50,000 times, making them two of the most highly cited papers in life sciences. In the 1990s, EST sequencing and microarray technology were developed and widely used. Many algorithms were created for gene expression data analysis. In the late 1990s, with the genome sequencing of the model organisms, gene prediction from the genome and whole genome alignment became hot areas for research. A good example is the hidden Markov model-based GenScan software developed by Chris Burge and Sam Carley in 1997. In 2002, Jim Kent published the BLAT algorithm in the journal Genome Research. BLAT can efficiently find the location of a gene on the reference genome. With the arrival of next-generation sequencing technologies in 2005, Sequence Read Archive (SRA) was created in 2007 to store the read sequences. It now has over 1,800 trillion nucleotides, doubling every 5 months. Because it was growing so rapidly, in 2011, NIH considered discontinuing SRA due to budget constraints. Fortunately, it changed its mind. Algorithms to analyze next-generation sequencing data more accurately and more efficiently are an important focus in bioinformatics at the time being and in the foreseeable future. So much for our review of the history. I hope that you can spend some time offline to read some of the classic papers to better appreciate the development of the field. Next, I'd like to mention that bioinformatics is not only an indispensable part of life science research, it has also helped drive the development of computational technologies such as neural network and genetic programming. Another good example is one of my fellow graduate students at Stanford University, Amit Singh, who, after learning about the BLAST algorithm in class, used the idea to develop a novel algorithm for wide area network optimization. The company he founded in 2000, Parabit, was later acquired by Juniper Network for $339 million US. I came to Stanford University to do my PhD in biomedical informatics in 1994. At the time, there were very few bioinformatics degree programs. In the 2003 review paper by Christos Ozanix and Alfonso Valencia, they recalled the early history of bioinformatics. They listed 10 of the earliest bioinformatics programs, including Stanford University, University of London, Boston University, etc. When I was doing my PhD, we all struggled with the lack of bioinformatics journals to submit our papers. At that time, there was only one journal dedicated to bioinformatics, which was called CatBios. Later, it was renamed Bioinformatics and matured under the leadership of Chris Zander and colleagues. Today, the situation has been much improved. The TAs for this MOOC, Meng Wang and Yang Ding, and I did a survey, and we found that today there are at least 19 bioinformatics journals, which are listed here. There are other journals that would publish a significant number of bioinformatics papers, such as nucleic acid research and genome research. We plotted the growth of the number of publications in PubMed and the number of bioinformatics publications in PubMed over the past 15 years. Figure A shows the growth of the number of all publications in PubMed since 1997. Figure B shows the growth of the number of bioinformatics publications in PubMed since 1997. As you can see, the latter has a steeper growth curve. If we calculate the percentage of bioinformatics publications among PubMed publications each year, you will see that the percentage has been increasing over the years from 0.1% in 1997 to 1.6% in 2012. This trend reflected the continued rapid growth of the field of bioinformatics. A field that is rapidly growing, driven by exponentially growing amount of data, is very exciting because it keeps creating new scientific questions and technical problems for research. The field is far from saturation, which is a good thing, especially for young people. You may ask, what about the history, trend, and current status of bioinformatics in China? In the next unit, I will share that with you. Here are a few questions for online discussion. We especially hope to hear your feedback on the last question. I look forward to meeting you online